Welcome, and welcome to those of you connected by Zoom from multiple campuses. Please remember, if you're connected by Zoom, to mute your microphone during the presentation. You can turn it on at the end and ask questions. So it's my pleasure today to introduce our visiting professor, Dr. Scott Selleck. Dr. Selleck earned his undergraduate degree in zoology, summa cum laude, from the University of Washington. And then he decided to transpose the University of Washington and Washington University, which confuses a lot of people in St. Louis. He moved to St. Louis to do his MD. Later did postdocs at Brandeis and at MIT. He joined the faculty at the University of Arizona and rose to the rank of associate professor with tenure there. That's better. Sorry about that. If you couldn't hear me in Zoom land, I'll start my introduction over, and we'll remind you, please, to mute your microphones at the distant side. So Dr. Selleck earned his undergraduate degree in zoology at the University of Washington, did his MD, PhD at Washington University, did postdocs at Brandeis and MIT, was on the faculty at the University of Arizona, where he rose to the rank of associate professor with tenure, and then was recruited to Minneapolis, where he had an endowed chair in pediatrics, developmental biology, and I think 2 other departments, Scott, as I recall, and ran the Minnesota Autism Initiative there. He was recruited from Minnesota to Penn State to be the chairman of the Department of Biochemistry and Molecular Biology about 7.5 years ago, the position that he holds now. Dr. Selleck has made a number of important discoveries in basic mechanisms of synapse formation using a fruit fly model that he's going to talk about in a research seminar tomorrow in Lansing. And today, he's going to talk about an important observation that he and others have made about the role of certain genetic changes in risk for autism, particularly copy number variants. And as you can see from his title today, he's going to talk about gene-environment interactions and their contribution to autism spectrum disorder, a study of copy number variation and air pollution exposures. So it's a real pleasure to have Scott here with us today, and thanks for being here, Scott.
Thank you. So this is working. Yeah, I can hear it. Thank you very much. Thanks for the kind introduction, and thanks for the invitation to come and meet you and tell you a little bit about some of our shared interests, I think. So this was the title that I provided to Keith when we're asking about it, but there's another title that will give you a little bit of an idea of some of the conclusions that I'm going to switch to, and that's this one, which is air pollution copy number variation interactions. And some of the results of this study, I think, have implications for understanding some mechanisms of autism and autism susceptibility. So this is sort of the alternative title that hopefully tells you a little bit about what we found in this gene and environment interaction study. So I know that most of you are probably familiar, but just for those who may not, this is sort of a brief history of this disorder. Of course, it's pretty long-standing now. And discovered and identified in the 1940s. But the primary hallmarks of the disorder remain, of course, which are impaired social development and language developmental problems, rigid and repetitive behaviors, and it remains true that the male to female ratio is very high. And what, of course, has changed over the intervening years is the prevalence, which by recent surveys are 1 in 56. And of course, there's a very active debate as to whether or not this is real changes in prevalence, or are these changes in our ability to ascertain and our awareness and ability to diagnose. And those debates are still raging, I think, to some extent. What are the causes? Well, we know that there is a pretty significant genetic component. The amount really depends on the method of study. I'd say 5 or 6 years ago, most people would say, well, it's 80% genetic. There have been some more recent studies on larger cohorts that would suggest that maybe the genetic contribution is less than that, but still significant. So I think that all told, some of the studies, the large, both twin studies from Sweden that were based on tens of thousands of individuals, have suggested that it's about 50-50, that environment, contributions, and genetics share equal roles in this disorder. And if you think about it, that actually kind of makes sense for a complex disorder, of which, of course, autism certainly is. So I want to take a few moments, though, to evaluate and review, well, what is our current power using the genetics and genomics tools that we have and those that have been developed? Where is that? How far has that come to allowing us to ascertain and identify a cause? And of course, a cause is a step to mechanism, which is a step to intervention, right? So I'm taking as an example a study that just came out in Nature Neuroscience, and this is from Steve Shearer's group at the University of Toronto, among others. It's a consortium of groups. And this team did whole genome sequencing of over 5,000 individuals. So best technology we have in determining sequence, right? Sequence everything, not just the expression, not just the mRNAs and the exomes, but everything. So there's probably $10 to $15 million of sequencing alone in this one paper. All right, so this is sort of the best we have with current technologies. So what did that generate from? So 5,000 family members, and I believe from this whole genome sequencing analysis, 2,620 cases of autism spectrum disorder. So after all was said and done, from that very detailed sequencing project, they were able to identify in 11.2% of the cases that they could say, aha, there is the genetic lesion that we have reasonable evidence is causative for this individual. Interestingly, of those 11%, 7.2% were copy number variations, that is, deletions or duplications, typically which involve more than one gene, right? So a good chunk of what they could put their finger on were probably multigenic events. But they also found in this sequencing, oh my God, how much variation is in our genomes between individuals. Just to give you an idea, per genome, they found on average 700,000 duplications, deletions, right? I mean, insertions or deletions of DNA. 700,000 per genome. If you look at single nucleotide variants, on average, the differences from their reference point, right, which was the reference genome, it's in the millions. 3 million or more on average. So there's an immense amount of variation in there that we don't know what it does. And it's pretty hard to figure out in combinations what that might do. So there is an immense amount of variation that is very challenging to study. And in fact, if you said, okay, let's take the hypothesis that interactions between 10 genes do this, to look at the impact of 10 variants together and in all kinds of combinations, pretty soon you run out of enough computer power in the world to assess those interactions. So it is a daunting problem, right? Which is why, of course, they're looking at individual variants at a time. Now, that's not the whole game in autism susceptibility and risk. There are known environmental factors. One of them is actually a protective one. We know that folate supplementation in children, or in mothers rather, in pregnant moms, reduces the risk of autism by about 2-fold. So it's not only protective for neural tube defects, it's protective for autism spectrum disorder as well. We know that maternal metabolic syndrome, gestational diabetes, obesity, those factors also are a risk factor for her child. We know that there are certain air pollution exposures that provide risk. And more recent work has shown that proximity of the pregnant mother and that child to agricultural areas where there's a lot of pesticide exposure is also a risk factor. So it's not as if genetics is all there is. But most of the studies, and it's really two different communities that do this work. They're the epidemiologists who do the exposures and the geneticists who take, you know, blood samples, DNA, and sequence. And they don't often engage one another. But we were interested in trying to correct that and make use of the expertise of both. But the studies that have been published thus far, both in the genetics and in environmental risks, largely do what? They examine individual genetic or environmental factors at a time. They don't look at combinations. It's not easy to look at combinations, but they basically have not been evaluated. And so the question remains, you know, if we can only account for, you know, 11.2% by sequencing their entire genome, for example, what about the other 89%? Is there gene-gene interaction or gene-environment interaction that's involved in those? And what might that tell us in terms of mechanism? So those are largely unapproached problems. So we don't know to what extent those interactions between genes or between genes and environment might contribute. And that is the fundamental question we set out to address. So again, our objective, why are we doing all this? Because we're trying to understand cause, and from there we are hoping we can ascertain some mechanism, and from there, intervention. And as I've spoken already this morning to many of you, I believe it's time. We do a lot of assessing, and we tell these families, your child has this copy number variant in 16p11.2. And there it stops, right? We don't offer them a treatment. We say this is the cause, but we don't often, and we don't know enough yet. You know, it's not that we don't want to, it's we don't know enough. But I can't tell you how badly I want us to do that, you know, to really strive at looking at the data we have and getting there. Because as we know, even if you can affect an issue with that child and family by 10%, make it better by 10%, it matters a lot to that family. So I'm bound and determined to try and contribute to that, I hope. So what we wanted to know in this study that I'm going to tell you about is, can we, first of all, measure the relative contributions of genetics and environment? In other words, how big are they, right? The geneticists say, well, it's all genetics. I'm exaggerating. And the environmental people say, well, it's going to end up being mostly environment. Well, what's the truth? Is there anyone who's actually measured the relative contributions, said quantitatively, can we make a comparison? We wanted to do that. And then we wanted to assess interactions and see if they would account for any of the susceptibility. Those were the two questions we had. So of course that requires that you get genetic and environmental data from the same set of children, and that is unusual. And what we had the advantage of is a study that was based at UC Davis by epidemiologists, and they had collected all sorts of environmental exposure information from those children and their families, and they had blood. And when I met Irva Hertz-Pachotto, who was the leader of that team, I said, can we do genetics on the blood samples you have collected? Because no one was. And she said yes. And so we created a team to do that. And so then we had some genetic information that they had already collected, and we're collecting a lot of environmental data. So that's what made it possible. And as you probably know, epidemiologists, they're really careful about controls. I mean, that's their shtick, right? When they get a matched control, they're not kidding. They mean a child that doesn't have the disorder, from the same geographical area, born at the same time, right? They don't just go to their freezer and go, well, I need some normal controls. I'm going to grab these samples that were done in an asthma study. I'm exaggerating. I'm kind of giving the geneticists a hard time. But I'm making the point that epidemiologists are, great controls are part and parcel of their profession, right? So we could take advantage of that. So we used those samples for some genetic analysis. So let me tell you the first study we did with that set. And what we were interested in was global copy number change. In other words, a lot of the variation between us, it's not just point mutations in single letters of the genetic code, but it's changes in whole words and even paragraphs, right? Where chunks of it are missing or duplicated or transposed. And there's a lot of variation of that sort. And we became really interested in that variation because the areas of the genome that display that variation, they are the hotspots for genetic change in our genome. So that seemed like a logical place to look because that's where action is happening. So we became really interested in deletions and duplications and the environments in the genome that created those and how they might contribute to this disorder on a global scale, not just an individual site, but across the genome. So we did this study, and we collaborated with the team at Davis and with Evan Eichler at University of Washington and some colleagues of mine at Penn State. And the question at the time of the study, we knew that autism had been found associated with an increased number of large copy number variants, deletions and duplications, but they were rare. So the perception was, okay, bad things happen every once in a while in these parts of the genome, and it's bad luck. And they are certainly contributing to autism, but they're relatively rare events. So the question that we asked was, maybe if you look genome-wide, they're not so rare. Maybe these kinds of things are happening broadly, and if they are they happening at a higher frequency in children with autism spectrum disorder? And the copy number variants that we are interested in, as I mentioned, they're in these genetically unstable regions of the genome. And these hotspots have particular sequence features that make them unstable, and I'm going to tell you a little bit about that. So our question was, if this genetic variation was found throughout the genome and not just in a few rare areas. And from the title of the manuscript that you can see there, of the paper, you can see that that's what we found, that there was globally an increased amount of copy number variation in those hotspots. I want to tell you a little bit about those hotspots. So this is a rather old paper, actually, from 2002, but it makes the point, I think, really graphically. So this is a map of the human genome, all the chromosomes, and what you see in purple are regions that have the sequence element that have been called segmental duplications. They and their relationship and the little connectors connect the ones that are related to one another, sequence related. You can see there are lots of them, right? And these are not little teeny sequences of 10 base pairs or 20 base pairs. These are on average at least 10,000 base pairs long. They're big. On average, they're copied maybe 6 times per genome. So they're not highly repetitive sequences. They're big, low repetitive sequences. And they can find related elements on the same chromosome and sometimes on other chromosomes. And those related sequences to one another promote instability. And there's lots of them. There's at least 170 of them. And at the time of this writing, which was when Evan Eichler was really, he made some immense contributions to this arena. And by the way, these segmental duplications, they're probably fundamental to human evolution. In fact, if you look at the structure of these in a mouse or a shark, they're simple. There are very few of them. They're shorter. And then you start going from old world monkeys to gorillas, chimps. There's an explosion of these things. And so Evan's hypothesis is that this sequence structure and its explosion in the hominid evolution line is absolutely essential to our evolution. Of course, as a species, we're only 200,000 years old. Compared to a shark, we're just an eye blink. And I dare say we'll probably be an eye blink, and the sharks will still be swimming around a long time after we've gotten into a fight with North Korea or something. So how do these duplications generate this instability? So this is a little cartoon to demonstrate that. So these are sequences that are pretty big. They're on average 10,000 base pairs or more. They're highly related. I mean, 95% or more sequence identity. And they're often separated on the same chromosome by, I don't know, 5 megabases or 50. And there are a modest number of them, as I mentioned. So how do those generate all these rearrangements? Well, you get errors in recombination is the answer. So here are a few examples of how they can mess things up. So here is one between two, homologous chromosomes, could be between two chromatids. And this yellow one and this pink one, these are segmental duplications related to one another. But if these two get out of alignment and there's enough identity between these two that they think they're brother and sister, so that they do a homologous recombination event, the outcome of that homologous recombination event is a duplication and a deletion. So that's what happens if this kind of non-allelic homologous recombination takes place between two chromatids, for example. If it's between two different chromosomes and you have them related, well then, these same kind of mispairings, right? They think they're identical. They're not, but they're close, and you get a homologous recombination event, and then you get translocations. If you get it within a chromosome, right? So this loops around, and they think they're partners, and you get a homologous recombination event. Now you get an inversion of that sequence. So this is just some of the ways where these long repeat elements that are scattered through our genome can do things. Okay. So back to the main point of this paper and so we wanted to know if the rearrangements in those segments of the genome in particular. So we focused our probes on those regions, but we also had some probes across the genome as a whole. So we really wanted to get a view of the entire genome. And what we found was that they have a higher level of copy number change across the genome. And we know that this made a difference in terms of the clinical outcomes because from the study that Irva and her team had done, they had a lot of phenotypic assessment, behavioral assessment of these children. And so we were able to say, okay, is the number of base pairs of deletion or duplication, what does it mean in terms of clinical outcome? And there was very significant negative correlations between the amount of copy number change, just base pairs of duplication total per genome, or base pairs of deletion, and the association with communication and socialization skills. So that led us to the study that was really what I want to drive home today, which is, all right, with that. So we have a genetic measure that seems to have a functional correlation with behavior that's genome-wide. And we have, as it turns out in that group of kids, exposure data as well. And we wanted to measure the relative contributions of these two. So we had that, we had case controls, and we had this copy number mapping data. So we could do all this. We had it done on the same individuals, and the first thing we looked at was air pollution exposures because one of Irva's collaborators, Heather Volk, had done the study and already published it. So what did she find? This was her paper published in the Journal of Medical Association, and basically what she showed was that a number of air pollution measures had an impact on the risk of autism. So let me just give you one table summary of her work. So here she's listing the risk for these different exposures. This is actually particulate matter of a certain size. This is another air pollutant particulate, a little bit bigger cutoff for the size. And then ozone, which is, of course, a nasty, highly reactive molecule. It's great to have it in the troposphere because it blocks UV. It's not so great at street level. And the street level ozone is not there to protect us. The street level ozone comes from air pollution, reaction of organic compounds with light to generate this nasty, highly reactive booger. Okay? So this is not, it's great up in the atmosphere, where it's protecting us from UV, not so good at street level, and nitrogen dioxide. So what she found in this paper is, in general, there were a number of environmental factors that increased your risk of autism by about twofold. Okay, double your risk. That was her number. So, and I should say, what is this, to be precise, she was asking if you had 2 standard deviations from the mean, that level of exposure to ozone or particulate matter, that level of exposure, what did that do to your risk? That was how she approached assessment and quantification of that risk. So we wanted to take a lead from that. So we want to do that same assessment for copy number variation. If you looked at 2 standard deviations from the mean of what was the average copy number load, what did you get? What was your risk? So we did that analysis, treating copy number variation as a continuous variable, which is what you do in the environmental exposures. And we started by assessing main effects. Simply, if you have this much duplication or deletion burden, what's your risk? And then we could evaluate the interaction. We had 305 individuals, not a huge sample size, but that's where we had both environmental data and our genetics data. And we also had the opportunity, of course, to measure not just main effects, but we could look at interactions. So we had copy number change in these simple measures, duplications, deletions, or adding them together, total copy number variation. And these were our factors that we had measured for environment. This is an estimated traffic-related pollution that Heather got from traffic patterns known in California, and the same ones that I mentioned, nitrogen dioxide, ozone, and particulates. Okay, so what was the interaction model? Well, the interaction model was that prediction of the phenotype, in this case ASD, would be a combination of your copy number variation burden, the environmental factor, and some interaction. And then we controlled for education level, sex of the child, ethnicity, things like that. The other model, the reduced model, would be that these interactions mean nothing, and that a perfectly robust predictor of susceptibility, of risk for autism, would come from simply your genetic risk, your environment risk added up, and then you control for the same factors to make sure you're not getting misled by differences in education level and sex, et cetera. We compared those models with a statistical test, excuse me, called the likelihood ratio. And that told us whether or not these interactions here had any bearing, which means did they have any predictive value. So let me tell you what the main effects of copy number burden are. And we did it actually in 2 cohorts. We weren't happy with just doing one, so we found a cohort that we could also assess of 588 children from the Simons Foundation collection. And the bottom line was that this risk of having this copy number variation was in the 2s, maybe a little bit higher than for some of the risks associated with the environmental factor, but it was certainly between 2 and 3. So main effects comparable between, again, this global measure of genetic change and some of the environmental factors. What about the interaction? So this is what surprised us. So this is the same thing. We're talking about 2 standard deviations, what the risk. And this number right here, this odds ratio, this is telling us that's the risk of the interaction alone between ozone and copy number burden. And this number stunned me. In other words, it's bigger than some of the principal factors that we're looking at. And of course, what was our question? Our question was, do interactions matter? Do we have any quantitative evidence that would suggest interactions matter, of which there was very little in the literature? And this told us there can be, and it can be significant. The other interactions were nowhere near this magnitude. In fact, this just gives you an idea of some of the different interactions we looked at. It's in the paper. And you can see that all the different combinations we looked at, whether it be duplications or total copy number variation, or what time period we're looking at, ozone comes up all the way. So that was the big one. Okay, now I'm going to show you the data without any fancy schmancy likelihood ratio testing and statistics and blah, blah, blah. I'm just going to show you the distribution of kids with autism broken down into quartiles. So ozone exposure, we've segregated into the lowest 25%, the middle 25 to 50, et cetera. And likewise, for the degree of genetic variation, the lowest amount of copy number change in quartiles. And then we simply said, okay, what proportion of the children in each of those quartiles get a diagnosis of autism? And we have nearly 50-50 representation of our matched controls and the children with autism. So nothing is happening, it's going to be 0.5. The ratio, you know, the percentage of children with autism diagnosis in those boxes is going to be 0.5 everywhere. So this is just the raw data. So this is what we got. Okay, so this is the quartile up here with the highest ozone exposure and the highest duplication burden. And the 82 out of 100 kids of that group get a diagnosis of autism. Okay? Down here at the lowest corner, 8 out of 100. If you go up these gradients, you know, there are trends. I mean, this is what the statistics has revealed to us, right? But this is just looking at the data. So you know, if you climb up in terms of copy number burden from the highest amount of ozone all the way up, this is, you go from 0.3 up to 0.8. Likewise over here, from the highest amount of duplicate, this is the exposure ozone of the risk of ozone. So it basically shows you that there is a gradation of these two factors, which was reassuring to me. Whenever I get a statistic, I actually like to see the relationship in the data without the statistics. Okay, so that work is just coming out in the next few weeks. I'm happy to say, and we got lots of questions about it. I think we spent a year in review, validating some of the epidemiologist questions about statistical methods and different interaction models and additive and multiple. I'm a simple geneticist. I'm going, oh my God, just publish the paper. But anyway, we finally got there with Irva's help at UC Davis. Okay, so the conclusions from that. These interactions matter. At least we know we have one case. They matter. They're not insignificant. They match main effects. So we need to think about them. And it could account for a considerable level of risk. So what about ozone? Why is it ozone? Why did the particulate matter? And we think that might have some mechanistic significance. I mean, because I have to say that our measures, the size of our samples, we're just getting the big skyscrapers that stand out in this analysis, right? With the number of children we had in our study and the sensitivity of our genetic measures and all that. So why did this one stand out? Well, as I mentioned, ozone is a very nasty molecule. So, what does ozone do? It contributes to the oxidation of molecules, and that's not good for you. Whether you're protein, DNA, lipid, if you have a lot of oxidation, it damages the function of those molecules. And there are lots of systems in our cells to counteract those effects. So is there evidence that elevated levels of oxidative stress has any role in autism whatsoever? Is there any evidence for that? Well, of course, there is. This is a paper from one of our colleagues, from Pat Levitt. And this paper, published a few years ago, looked at the levels of oxygenated lipids that you could get from blood samples from children with autism. And particularly, he was interested in those that have the behavioral elements of ASD, and they also have GI symptoms. Those children had the highest level of lipid oxygenation from the blood samples he got. But even the children without the GI disturbance, the children with autism also had significantly elevated levels of oxygenated lipids in their blood, which says they have some oxidative stress level. And this is only one study of many. This is, of course, focused on peripheral, not in the brain. This is just using the blood as a measure of what are these children exposed to and is it significant. But here's a review article that just came out not too long ago, which lists many of them. And one of the reasons I picked it was because there is a functional connection between oxidative stress and mitochondria. Why is that? Because mitochondria, that's what they do for a living. They oxidize and reduce, right? They take, that's how we get energy So oxidation is sort of a two-edged sword. It's really great. You get lots of ATP when you oxidize carbohydrates, but you also have lots of reactive molecules. And if those electrons escape the electron transport chain in mitochondria, those guys get away, they do damage. They are, in fact, reactive oxygen species. So there's a very sophisticated system in cells of identifying mitochondria that are not working well, and the cell degrades them. There's an intracellular system that engulfs those mitochondria that aren't working properly, and they get degraded into fundamental components of lipids, amino acids, et cetera. They're chewed up and put in the garbage disposal, okay? So there's a connection between oxidative stress and mitochondrial function. So what are the sources of? Well, I've sort of mentioned some of them. First of all, there are exogenous sources, right? Ultraviolet light, environmental toxins, et cetera. And then there are endogenous sources of reactive oxygenation species also, mitochondria being principal among them. But there are endogenous, and we have antioxidant defenses, thank God, or else we'd be all turned into free radicals. So, what cellular process connects these things? Mitochondrial function and oxidative stress responses. And the answer to that is autophagy. So autophagy is a cellular system, and it literally means self-eating. It was identified initially in studies of nutrient stress and starvation. So when cells are not getting any nutrients and they need building blocks, what they do is they chew up existing things, turn them into building blocks, and synthesize them, okay? So the induction of this process was discovered as a response to nutritional stress, all right? Self-eating. And literally, a double membrane vesicle is formed, and it reaches out like a cup, and eventually, for example, it'll surround a mitochondrion. And then into that membrane-lined vesicle, it fuses with lysosomes, which is where all the degradative enzymes are for cells, and that whole thing is chewed up, and the building blocks used again, all right? But that's not the only thing that this process does. It also gets rid of misfolded proteins. They're not good for cells, you know, whether that be senilins, whether that be alpha-synuclein in Parkinson's disease. This is the system that is tantamount to saying, okay, I've got this out-of-solution aggregate, and we have to get rid of it. This is one of the systems that does it. So it doesn't just play a role in nutritional stress and getting goodies to build with, but it's there to clean up the world. It's your sweeper in cells. It's also involved in reorganizing synapses, right? When synapses change in development, a lot of the proteins that lock a synapse into place, this system will target some of those proteins that are involved in locking that synapse into place, degrading those so that the synapse can be rebuilt or expanded or changed. So autophagy plays so many fundamental roles. You can tell I'm kind of interested in it. So this is another cartoon. It's sort of the cell sweeper. It regulates issues to do with aging and programmed cell death, defense against pathogens, adaptation to starvation where it was first identified, and elimination of aggregated proteins and defective organelles like mitochondria. So if you don't have autophagy working, you're going to accumulate mitochondria that are going to deliver reactive oxygen species, okay? All right, so how does all this relate? Is there any evidence that this process that I'm blithering about plays a role in autism? Another rhetorical question. This is a beautiful paper, and David Sulzer, this is the paper I was telling you about at breakfast. This is a really amazing paper in Neuron a few years ago. And the title sort of says it all. It says that loss of mTOR-dependent macroautophagy. So as it turns out, the regulation of autophagy is mediated by signaling pathways that we know quite a bit about, and they have been implicated in autism. And If you don't have that process working correctly, they found deficits in synaptic pruning. And this paper, I highly recommend it to you. It's beautiful. It has both analysis of the structure of synapses in human brains in autistic children. I think they had 10 children with autism and 10 controls that they could look at. And the bottom line was, in those comparison, there are a lot of synapses which are made, and in normal development, they get pruned back. Right? So you have an excess of these connections, and then you prune them so your nervous system function is appropriate. Right? So you start with an expanse, and then you prune. That pruning is not happening in those kids. The question is, what is the basis of the pruning? What is the cellular machinery that governs that pruning and eliminating synapses that you don't want and, in fact, are confusing the picture? As it turns out, at least one of them, they would argue in this paper, it's autophagy. That cellular process of identifying proteins for destruction, getting rid of them, that process in a mouse model of autism they use, which is a TSC heterozygous mouse, that was autophagy dependent. Okay, so how am I doing on time? Okay-ish? Okay, so I'm going to tell you a little bit of a story from our Drosophila, our fruit fly work that relates to this. Quite independent of us, working on this problem and what does ozone have to do with it and what's the evidence that this might be an oxidation exposure, et cetera, we were doing some basic genetics of synapse assembly in the fruit fly, looking for genes that were essential for building a synapse. Okay? So this was a genetic screen for genes that were required for the postsynaptic specializations, right? So there's the presynaptic cell and the postsynaptic cell, and there's some very unique specializations of the postsynaptic cell that make sure that this chemical communication central to all the nervous system works properly. So we did this genetic screen, and I'll tell you, so here's sort of a cartoon of it. So here is a synapse with the presynaptic cell releasing these vesicles with neurotransmitter, and then the postsynaptic cell with its receptors, and of course they mediate the movement of ions across membranes, and a chemical signal is converted to an electrical one. Now, as it turns out, so you might say, well, you're studying autism-related things in a fruit fly, how does that work? Well, one of the ways it works is that this synapse that we're studying, its neurotransmitter is glutamate, and all the components of signaling and receptors and neurotransmitter and its synthesis that occur in our brains, which is an excitatory synapse. They're found at this synapse. It turns out the neuromuscular junction, right, where the neuron comes, makes contact with the muscle, and when synaptic transmission happens, you get contraction. That synapse has glutamate as a neurotransmitter. So that's handy. Okay, and here's sort of what it looks like in real life. So this is the, I don't like to use this word, but this is a maggot, right? We don't talk about maggots. They're larvae. But in the vernacular, common vernacular, this is the little fruit fly maggot that's crawling around before it becomes a pupa and before it becomes a fly, right? Just like a butterfly, same general body plan development. So if you slice one of those up and splay it out and pin it down, and you take out the guts, and you look at the muscles, you have this beautiful segmentally repeated muscle structure where all these synapses occur. So this is what it looks like when you fillet it. And this little thing up here is what remains of the brain. So these are strap muscles for each. There's an abdominal segment, there's one. And if you look closely and you stain with antibodies that recognize neurons, you can actually see the neuron termini on these muscles. So literally, this is one cell. From here to here, that's one muscle cell. Lots of nuclei in one cell, but one muscle cell. And this is what the synapse looks at a higher level of magnification. And this green stuff here, it's actually just a tag of membrane. Because the postsynaptic cell at the muscle, where the motor neuron terminal fits in there, this membrane here, it folds up into all kinds of intricate fingers. And there's lots of membrane there. So if you just take a fluorescent tag that goes into a gene that's in lipid, goes into a membrane, there's just tons of it right there. So that's all the postsynaptic special, all the membrane that's associated with those infoldings. And this is kind of what it looks like in cartoon form. So here is that, the motor neuron bouton embedded in the surface of the muscle cell, and the muscle cell membrane goes through all these infolds and contortions as the synapse grows in development. And so this is adaptive to the muscle cell getting bigger, the larva growing, etc. So it's that morphological feature we looked at. And those specializations, even though they're infoldings like that, they're really functionally equivalent to the dendritic spines in the brain that I was talking about. So these are the dendritic spines. These are the things, here's a neuron, and all these little blebs are potential receptive fields. These little stalks and heads are called dendritic spines, and that's where synapses take place. And it's the pruning of those that was found to be deficient in children with ASD. Okay, but these little dendritic spines are the same thing, postsynaptic specialization. So we started this hoping that the genetics and molecular biology of a simple synapse in a Drosophila larva would have functional correlates to this. And there are many examples of it that I don't have time to tell you about. But what do we find out? Well, these are these specializations. This is an example of one gene that came out of this genetic screen. So these little green buttons, they're actually, in each one of those buttons is going to be a motor neuron terminal, okay? And those green buttons are the infoldings of the membrane that represent that postsynaptic specialization to make sure all that synaptic transmission takes place normally. And this was one of the genes that we identified in our screen. It's a gene called acinus. And we got this kind of structure, very diffuse, all over the place, not looking very good. Well, it turns out that that is an autophagy functioning gene. So one of my colleagues at UT Southwestern, Helmut Kramer, whom I was a postdoc with, he has spent a lot of time and energy in the gene that he came up with in looking at endocytic traffic and movements of membranes and cells. This gene, acinus, came up in his screen. So that suggested to us that autophagy as a process was important for postsynaptic development. And so we've looked at other components, and lo and behold, here's the nice little bouton shape that you should get in a normal. There's the strap muscle here, and these are all those dense infoldings of membrane. So this is knocking down a completely different gene that's also essential for this cellular process of autophagy. And we get the same phenotype, this diffuse, ugly, non-happy postsynaptic feature. Okay, I'm going to try and put all these pieces into one framework. So is there a common pathway for all these contributing factors that we've gotten from our environmental studies of genetics and air pollutant exposure, many other people's work, and some of our analysis that autophagy as a process is important for remodeling synapses too? I think there is. This is the pathway. So I'm going to point out a couple things. First of all, this is a growth factor pathway, or pathways, that has been implicated in autism in many different terms. You'll recognize, I'm sure, many of the elements. Here are the genes for tuberous sclerosis complex, okay? We know those have a role. One that's not listed here that is still important is PTEN. So there's many components of this signaling pathway which are implicated in autism susceptibility in humans. What does it do? Well, one of the major things it does is inhibit autophagy. TOR, the target of rapamycin, through which a lot of this signaling is funneled. That kinase is the big daddy regulator of autophagy, and it suppresses it. So let's say you have a mutation in TSC. You turn this signaling on. TOR is hyperactive, and you will suppress this. And not having enough of this pathway of recycling your mitochondria, getting rid of aggregated proteins, that is not going to be good for you, okay? So, and this relates to the ozone exposure. If you have heightened level of oxidative stress and autophagy is not getting rid of your mitochondria, that's not going to be good either. So there is, I think, some conceptual connection between many of these different features. So here is some of the data that ties it together. So we know that growth factor signaling through that pathway to TOR matters. Well, what's the relationship? Well, here's some of the genes implicated in autism that we know are involved, and what do? They put a brake on this thing. And these are PTEN, TSC1, NF1. Those genes have an inhibitory effect on TOR, which means you would allow autophagy to work, right? Well, what else? We know that maternal obesity, metabolic condition, and even imprinted genes are implicated in autism. Guess what? They all promote signaling through this pathway. What else? Well, we know this is an important thing, and these are the different biological processes that autophagy regulates. It actually regulates DNA repair. So, in our copy number variation analysis, we found they have more genetic susceptibility. They have more genetic change. Is it possible that autophagy plays a role in susceptibility to that? We know that synapse remodeling is an important part of autophagy by a number of different studies, and it's associated with change with autism. We know that it removes mitochondria, and we know that increased levels of reactive oxygen species are found in children with autism. And we know that it regulates protein turnover, and there are mutations in the protein turnover apparatus that also are involved in autism. So that's sort of trying to distill these variant elements into something that might make sense. So it suggests this mechanism that's involved, many different genes affect this pathway. It's been suggested by others, because of that genetic evidence, that maybe TOR inhibitors are an intelligent thing to try for treating autism. And they are being used to treat children with tuberous sclerosis complex. And there's a clinical trial now with children that have PTEN mutations. And of course, those are very rational because we know that those children have effects in those pathways that ultimately result in the abnormal activation of TOR and the abnormal suppression of autophagy. So this is a paper that just came out, essentially an opinion piece almost, from Atsushi Sato, who said we should really think about this. And his arguments were based on the involvement of that pathway, not the autophagy link. And I think the evidence that's come out in the last couple of years on autophagy make even a more compelling case. But I wanted to point out that I don't want you to get the impression that I'm the only person that said, hey, maybe we should think about this. There are a lot of people who've been thinking about it. But those drugs, and they are available, of course, they are rapamycin derivatives. They are the drugs that made transplantation of organs possible, right? And some of them get across the blood-brain barrier into the brain, all right? Okay. That's my take. That's what I think is one area that we could consider. Is this pathway, is all this different genetic environmental data telling us that this pathway is something we can, perhaps might be involved in a substantial proportion of children with autism? Maybe not just 11.2% with totally different genetic lesions. So I think this is the kind of thing that maybe we can begin to move into trying some of these approaches to helping these children. So, of course, this is not done, this is done by a whole group of folks, and I just want to acknowledge some of them. My professor collaborators in this project, Marilyn Ritchie at Penn State, Duk-Yun Kim was the postdoc who really took that gene interaction, environment interaction paper forward, as well as a new faculty member at Penn State, Santosh Girirajan. Irva, of course, is the head of the team at UC Davis. Evan Eichler contributed immense expertise to the copy number analysis. Heather Vogt, who did all the air pollution related work. And these are some of the members of my lab that have contributed, Jay and Na, and I should add that these are several of these are undergraduates who were involved in the computational stuff and some of the fly work. And with that, I will close, and thank you very much.
Questions?
Yes.
I'll ask one.
Okay.
How about effects of epigenetic effects of ozone, separate from that? Does ozone have direct epigenetic effects, or is it really interfering with other epigenetic?
Well, I mean, the broad answer to your question is, of course, these things can very, very well affect epigenetic factors. I don't actually know the direct influence of ozone on epigenetic transmission mechanisms, but it's something to really pay attention to, because it's just another form of, I look at it as inheritance that's not as long, but they're definitely heritable events, and Janine LaSalle's work at UC Davis has really highlighted that. So she's been very interested in documenting some of the epigenetic changes in the 15q region. And she also wrote a beautiful review lately describing how many genes involved in cancer are implicated in autism. And I think she came up with 40. It's in Trends in Genetics last year. But epigenetics, it is definitely as viable a component of these changes as any other. And there are fewer people like Janine really tackling it. But it clearly has an impact. How ozone influences it, I just don't know. Yes, Eric.
So, again, fascinating work for your talk. Two questions. Do you have any idea what proportion of the copy number changes are de novo in these children? And at the end of the day, do you think that the copy number changes will turn out to be driving for activation, or will it be the other way around?
I wish I could tell you. Yeah.
Can you repeat the question?
Yeah, so let me repeat the question. The question was, the copy number changes that I've described, how many of those were inherited from the parents or were new events in the affected child, that is, de novo events? And then the second question was, given those genetic changes, do I think they are the ones affecting directly the signaling system I've described and contributing to susceptibility in that manner? Unfortunately, we didn't have the money to do the parents to address the first question, which I would have loved to have known, what degree are inherited and what degree are de novo. With regard to the second question, we don't, again, we're limited by sample size. So if we look at the genes that are affected by the variants in our group, if you just look at it raw, you don't see any direct indication that, yes, these are events that are going to affect TOR signaling. So on the face of that, you'd say, oh, well, maybe this genetic change is a consequence, not a driving factor. But we've recently done an analysis where we look at and it's not published yet, where we look at the genes affected by the variants. And we categorize them based on various terms, Go terms, functional terms, et cetera. And then we look at their association with autism. And as a comparable model to get statistical comparisons, we will randomize the autism diagnosis. And we'll do that 1,000 times. And we'll see if the distributions of genes are affected compared to our randomization and just see what drops out. And when we do that analysis, we get things that involve protein turnover at pretty significant rates. We get some other things, but among the top hits of that modest analysis are things that affect ubiquitination, protein turnover, membrane trafficking, or some events. So that study, we're sort of we've developed, I think, a useful method to assess copy number variants in this way, but we're a little worried about publishing something with that number. We really need to have it 3 times bigger. But I think, I guess what I'm trying to say is, done in that way, there's so many different connections, right? So you can influence that pathway through so many different ways, right? And There are databases on, as you know, protein-protein interaction databases that can tell you what is the cast of characters that this one component hangs out with that might tell us something about the functional consequences. So the pure genetic lesion, it's not always trivial to figure out what is the neighborhood in which it's working. So I don't have any real definitive ways of telling you whether or not these events are a consequence or whether or not they're drivers.
We'll stay tuned.
Yeah, I hope so. Getting a yes.
Since autophagy
Yeah.
is a continuing process, not just developmental, you're suggesting that these rapamycin derivatives can provide treatment for an adult person with autism.
Well, that's a very your question harkens on if you had a good mechanism and you had a good way to intervene, when can you intervene and have an impact?
So many of us worry that it's strictly occurs at the developmental stage and
Yes.
there's nothing you can do.
Yes.
Thereafter, there are certain things you can do.
Well, I mean, the bottom line answer to your question is we don't know. I'd say the likelihood of interventions late, the likelihood of them having a big impact is less than developmentally. So let me say a few things about the patterning, for example, of this pruning of synapses that takes place. So in normal development, as the brain grows, you expand the number of these synapses dramatically, and then you prune them. And that pruning occurs from age, from birth. Well, actually, the expansion goes to like age 1 or 2, right? And then from then, they begin to prune them. And so that pruning goes from like age 2 to 12 or something. It's a pretty big window. So one might say that, okay, if that's really critical, and regulation of TOR via drugs like rapamycin is useful, you could argue that anywhere within that window might have a positive effect. Whether or not there's some process of that nature that occurs in the adult that still might be useful, we just don't know. Or even, let's say you have a deficit in being able to turn over mitochondria. What impact does that have on you throughout your whole life? So I think the likelihood of successful intervention is the earlier the better. But where that cutoff is, I don't think it's going to be finite, right? And so I would love to see a clinical study of these drugs, as you can tell, right? So the clinical review boards are going to ask, what is your evidence that this drug is targeting a mechanism in that patient and that individual? Because we're not going to let you just put in an immunosuppressive drug into a child just willy-nilly because you've got a groovy little hypothesis. So that means we need markers that these biochemical and molecular events are affected in that individual to screen them into the study, for example. But that's not impossible. So what we need to do is we need to have molecular signatures, perhaps, of very high levels of reactive oxygen species in their blood, or markers that this process of autophagy is down in the dumps and isn't where it should be. With that rationale, we might be able to make a compelling argument that these are worth testing. Now, they're already being tested in children with tuberous sclerosis complex, and a lot of those children have autism diagnosis. Now, most of those clinical trials have not been directed at watching behavior. They've been directed at watching the benign tumors that are associated with tuberous sclerosis complex. So there have been so the good news is they have been tested. They're not horribly toxic. Their dosing schedules are known, and there's some that penetrate the brain. So there is data that is available to help guide things. The tricky part, I think, would be identifying the children that you can reasonably justify that kind of trial on. But I think that's doable too. So that's one of the things we're working on, is identifying a metabolomics profile that will say, aha, lots of reactive oxygen species, autophagy is not doing well, and what are the molecular signatures of that we can apply? Yes?
Yeah, very nice, Scott. So a couple questions. First, comment to Jim's question. So Mustafa Sahin has done Purkinje cells,
Yeah.
which are like best cells of the brain, fixed tuberous sclerosis, right? And got learning to have a cognitive disability.
Mm-hmm.
These were in adult mice, more or less, or at least not post-embryonic.
Okay.
Right? So the treatment, well, I believe, like you, that earlier you intervene, the better. It holds out for even, through the lifespan,
Right.
possibility. But I love the idea of a common pathway towards autism. But then how do you juxtapose that to the comment? It's absolutely true that we met one autistic kid,
Right.
and the heterogeneity of the issue. And then you go and find 10 other theories
Yep.
of autism. We were talking about the excitation inhibition
Yep.
one. There's the one on microcolumns, and there's the, you can dredge up lots of them.
Yep.
So do you have faith that this common pathway can be the grand aggregator of all these different ideas?
Well, I have a degree of faith. I will tell you, philosophically and scientifically, I'm a reductionist. Okay, I believe I am a bit skeptical that some of these more neurophysiological models. That's not to say that those don't happen in individuals, that they are relevant. The question is, how do they get there? I think autism is fundamentally a disorder of neurodevelopment, and the phenotypic consequences are on the two phenotypes in humans that we know the best: language and social interaction. And there's 1,000 ways to get there. So yes, it seems in complete contrast, but I do think it's possible that a good proportion of those have lesions that, at all the myriads of ways that this fundamental signaling pathway is affected, that it might have some impact. I don't know how much. I just want to have an impact. And so if we can, as I said, if we can improve the expression of the phenotype or of the language disorders or social interaction, if we can back that off by 10% or whatever margin in 30% of the, that's a victory. And I think it's time for us as a community to do that, again, within the guidance of we don't harm anyone. So I really don't. Part of what I described to you is a dream, right? This is my dream, that this will be true for some substantial proportion. I think there's enough evidence from all these different domains of our community that suggest it's not crazy, that they might be connected. So I'm cautiously optimistic, I guess, but I think it's worth trying, is what I'm saying. Yes.
I heard Keith draw his breath that was about to say, I'm sure we need to leave.
Oh.
So we've heard mouse models mentioned, and Dr. Jan was here a couple weeks ago. She has a mouse model. And as you know from our morning, I'm no advocate of mice. But do you have any plans to try some of the rapamycin derivatives in these mice models, which may or may not be appropriate to your hypothesis, but is that in the works?
I would love to try it. Obviously, the rapamycin treatments have been tested in some of those mouse models for effective pruning of synapses, and it works. It fixes that pruning thing. But there are other models that would be useful, I think. As I was telling you, my colleague Santosh Gururajan is taking these genes in flies, and what does he get with the majority of autism susceptibility genes? He gets growth problems. So that's actually, you can feed rapamycin to a fruit fly and it works perfectly fine. It inhibits TOR to beat the band. So that conservation of signaling, you can test that even in a fruit fly too. So yeah.
Thanks very much.
Oh, thank you. Thank you.